Please welcome Sophie Tritschler, Helmholtz Zentrum, München. A single-cell lens on tissue failure and drug response in diabetes.
500 million people, this is the number that is expected to have diabetes in 10 years. Thus, we urgently need novel treatment options. When we eat one of these gingerbread hearts, our blood sugar levels increase and insulin is produced in the cells in the pancreas. In diabetes, insulin's production is insufficient due to cell death, but also because cells fail and become dysfunctional. A promising alternative treatment strategy is now to target these cells and make them functional again. We have tested several drug candidates along these lines in a large-scale study in mouse. In a healthy pancreatic tissue section, we see a high production of insulin, here stained in red. In diabetes, this is gone. And amazingly, we now find several drugs that actually restore cell function and insulin production. But two important questions remain. Why do cells fail in diabetes? And how did our drugs work? Basically, how was cell function recovered? Tissue failure is a continuous process, and the individual cells fail at a different time and at different speed during disease progression. Thus, in a diabetic pancreas, we do not just have a single diabetic cell, but a whole spectrum of cellular states. And if we measure these individual states by single-cell genomics, we can actually reconstruct a path from a healthy to a diabetic cells by simply ordering these cells based on their similarity. And as we have measured the expression profile of each individual cell, we can now also explore gene expression over time. In real, we have isolated cells from healthy and diabetic tissue, we have measured the gene expression profiles, and we then reconstructed the path of cell failure basically from a healthy diabetic cell. By this, we can see which pathways drive the cell failure and make cells dysfunctional. And how can we now revert this process by drug treatment? When we compare the profiles of the distinct drugs that actually restored insulin production, we find that the pathways that trigger cell function, that reactivate cell function. But in addition, when we look at the difference between the response profiles, we can also resolve the mechanism of action of each of the drugs. So I've shown you that from a list of candidates, we have learned in a mouse study which are effective to restore insulin production. We use single-cell genomics to understand why cells fail in diabetes and how they respond to treatment. But we can even go one step further. We can learn in mouse and actually translate these findings to human using AI. We can learn a deep learning model and use this model to predict drug response in human and thereby rule out candidates that are not effective.